This video is about eukaryotic transcription. Eukaryotic transcription is inferred as a process in which the eukaryotic cells replicate the data in a strand of DNA and copy the genetic knowledge into a new molecule of RNA. Eukaryotic transcription is referred to as a first step towards gene expression in which some segments of DNA are traced into RNA by a specific enzyme called RNA polymerase. This results in forming an antiparallel RNA fiber known as primary transcript. What is that? Primary transcript. And let's learn about the procedure of eukaryotic transcription. Transcription in eukaryotes take place in the following manner. Transcription is the formation of RNA over the template of DNA. It creates single-stranded RNA which has coded information similar to the sense or coding strand of DNA, with the exception that T is replaced by U. T is replaced by U. The DNA strand, which functions as a template for RNA synthesis, is called template or antisense strand. The segment of antisense DNA that takes part in transcription is called the transcriptional unit. It may consist of one or more histones or genes. Each transcription unit has a promoter region, generally in the beginning, and terminator region where transcription ends. What is the mode of transcription? The enzyme taking part in transcription is called RNA polymerase. There is single RNA polymerase RNAP in case of prokaryotes. We learned about this prokaryotes with the help of a video which early access to the video is there. So I just posted that video earlier. So eukaryotes have three types of RNA polymerase. RNA polymerase 1 for synthesis of ribosomal RNAs except 5S. DNA RNA polymerase 2 for mRNA and many snRNAs, along with RNA polymerase 3 for tRNA, 5S RNA and some snRNAs. Prokaryotic and RNA polymerase has a sigma factor for recognizing the start signal of the promoter region. The remaining part of RNA polymerase is called core enzyme. In eukaryotes, separate protein factors take part in recognition and initiation. They are called transcription factors TF2, TF3. Promoter region of the transcription unit has a separate recognition site and polymerase binding site. Terminator region of the transcription unit has either palindromic sequence or poly-A sequence. Termination of transcription requires a separate termination factor called Rho factor. The various steps in transcription are as follows. Activation of ribonucleotides. 4 types of ribonucleotides take part in the synthesis of RNA over DNA. They are AMP, GMP, UMP, and CMP. The nucleotides are available in the nucleoplasm. Before their incorporation, the nucleotides are converted into active weight through phosphorylation. It produces triphosphates. Energy, phosphate, and enzyme phosphorylates are required. Then DNA template. Only one DNA strand functions as template strand. It is also called as an antisense strand. So DNA template, only one DNA strand functions as a template strand. It is also called an antisense strand. The unit of transcription begins with a promoter and ends in a terminator. The separation of template strand does not require specific chemicals as in case of DNA replication. Instead, the core enzyme of RNA polymerase travels along the template strand with the help of protein factor called NUS-A protein. Then initiation. RNA polymerase reaches the promoter region. Sigma factor recognizes the promoter region. In eukaryotes, there are separate transcription factors for recognition of promoter regions. As soon as the enzyme RNA polymerase gets attached to the template DNA of the transcription units begin to unzip. RNA polymerase reaches the promoter region. Sigma factor recognizes the promoter region. In eukaryotes, there are separate transcription factors for recognition of promoter regions. As soon as the enzyme RNA polymerase gets attached The template DNA of the transcription unit begins to unzip. Then base pairing will be taking place. The activated phosphorylated ribonucleotides come to lie opposite complementary nitrogen bases of the template strand. A opposite T, U opposite A, G opposite C, and C opposite G. With the help of enzyme phosphatase, the extra phosphate radicals of nucleotides are hydrolyzed. It releases energy, which is helpful in establishing temporary bonds between complementary base pairs. Then formation of RNA chain elongation. The core enzyme, with the help of energy and Mg2 plus, builds phosphodized bonds between adjacent ribonucleotides, forming the RNA chain. As the enzyme moves along the DNA template, the RNA chain elongates. Synthesis continues till the enzyme reaches the termination region. In the termination region, RNA polymerase is separated from the DNA template by means of Rho factor and NESG. Then chain separation or termination. Rho factor has ATPase activity. This separates RNA polymerase as well as newly built RNA strand. As soon as the RNA strand separates, the sense and antisense strands of DNA re-establish hydrogen bonds between their complementary base pairs. The duplex nature is restored. Post-transcriptional changes. Freshly synthesized RNA strand is called the primary transcript. It is often bigger than the functional RNA. The various modifications which occur in primary transcript are as follows. Cleavage. A bigger primary transcript is often cleaved to form RNA of desired length. For example, the primary transcript of rRNA is 45S. It undergoes cleavage with the help of RNA enzyme ribonuclease P. Then splicing. Most of the eukaryotic mRNAs contain non-coding segments called introns or intervening sequence. They are removed. The process of removal of introns through cutting and joining the essential sequences or exons are called splicing. This is carried out with the help of snRNPs. The later are formed by association of proteins with small nuclear RNAs, snRNAs. snRNP binds to 5' end of the intron while other attaches to 3' end of the intron. A spliceosome develops there. With the help of energy from ATP, cuts are made at both 5' and 3' ends of introns. This releases the introns. The ends of the adjacent exons are joined together to produce processed RNA. Then terminal additions. The additions are made at the ends of newly formed RNAs for developing facility to recognize the area of their action. For example, CCA is added at the 3' end of tRNA, which is the area of attachment of amino acids. mRNA develops a cap at its 5' end. The cap is 7-methyl guanosine, 7-mg. It develops at the 3' end and consists of repeated A nucleotides. Then nucleotide modifications. Nucleotides are modified post-transcriptionally to perform different functions. Maximum modifications occur in tRNA. They include methylation, ethylation, deamination. It produces such chemicals as dehydrouracil, cerouracil, methyl cytosine, and eosin. The detailed explanation of all these points about eukaryotic transcription will be included in the next video. Thank you for listening. So this will be the part A. Later, we are going to learn about the part B. The detailed explanation of all these points like initiation, elongation, and termination. Thank you.